What is the minimum number of genes a bacterium needs to live? In other words, what is the bare minimum for a bacterium to survive? Which genes are the most important? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Edward Kejnovsky, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. To create a human being, approximately 21,000 genes are needed. A similar number of genes can be found in many animals and plants. In eukaryotes, genes make up only a tiny part of the genome. But it's different with bacteria. Their genomes have been pushed by evolution toward different strategies than those of eukaryotes. They had to reproduce as quickly as possible and survive even in unfavorable environments. Everything unnecessary was therefore removed from their genome. A bacterium simply couldn't afford to copy unnecessary DNA in its genome. That would slow things down and would also be uneconomical. They have to get by with just hundreds or at most thousands of genes. The question of the minimal genome, or the minimal number of genes in the genome of an autonomously living organism, is one of the most provocative questions in biology. It even has a certain philosophical significance. The smallest genome among known organisms belongs to the parasitic bacterium Mycoplasma genitalium, which lives in the reproductive organs and the respiratory tract of primates. It manages with just 470 genes. It can afford this because it lives in close association with a warm-blooded host, to whom it delegates a number of metabolic functions and who provides the bacterium with a constant environment. Do we really have in our hands the bacterium with the smallest genome? We realize that we know only a small part of the bacterial world. Most species are hidden from us, let alone their genomes. That is why it is quite likely that somewhere on Earth there are bacteria with even smaller genomes. The bacterium Pelagibacter ubique demonstrates just how limited our knowledge of the world is. It has one of the smallest genomes with only 1,354 genes. This bacterium may be the most abundant bacterium on Earth. It occupies almost the entire water column of all the oceans. It's almost unbelievable that it was only discovered in 2002, which is quite recent. And that's despite the fact that the total mass of all bacteria of this species exceeds the mass of all the fish in the oceans. Scientists were interested in what is the smallest number of genes sufficient to encode bacterial life and which genes those are. Nowadays, the entire genomes of thousands of bacteria have already been sequenced, so it's possible to look for genes that are common to all bacteria. Those are probably the most important of all. But scientists went even further. They conducted elegant experiments in which they systematically deactivated individual genes. In their experiments, they used both the bacterium with the smallest genome, Mycoplasma genitalium, as well as bacteria with larger genomes, such as Bacillus subtilis. In both cases, they arrived at a similar number of genes with which the bacteria were still able to survive. Naturally, the minimum number of genes also depended on the environment in which the bacteria were growing. In a nutrient-rich environment, the bacteria needed fewer genes. For the bacterium Bacillus subtilis, scientists determined there were 261 absolutely essential genes. For Mycoplasma genitalium, earlier estimates put the number at 265 genes, but more recent experiments suggest a higher count, specifically 382 genes. If we were to look at the list of essential genes, we would find genes for the production of DNA and RNA molecules, for the construction of ribosomes and everything necessary for protein synthesis, genes important for building cell membranes and cell walls, as well as genes that control the basic processes of cellular energetics. The function of approximately 100 essential genes in Mycoplasma genitalium is unknown. That's all for today. In the next episode, we'll talk about how scientists are trying to create an artificial genome. Let's discuss what has been encoded into the artificial bacterial genome. I'm looking forward to seeing you. If you enjoy my storytelling, I'd appreciate a like. And if you'd like me to continue talking about evolution and genetics, please subscribe, which will help these videos reach more people. I really appreciate your support. Thank you, and bye-bye.